have a question. Why did you choose lamps as a control?
These lamps are widely used in greenhouses on Earth and are known to give a sufficient yield of biomass.
Done? I don't need to run with times.
Do we have time for several questions? You said you had a ratio of 7 to 1 of the red and the blue. Was that ratio of photosynthetic photon flux, so there would be in the 100 micromole treatment, 70 micromoles of red and 30 of blue?
Yes, it was about that.
So it's based on photon flux?
Yes, it is photon flux.
Did you make any measurements of chlorophyll and leaf morphology? I noticed that some of the treatments, the leaves were more elongated, and some they were more compact.
In the experiments with assemblies based on white LEDs, we have not yet done any measurements of pigments or other parameters of the photosynthetic apparatus. In the experiment with red and blue LEDs, we did a measurement of chlorophyll content and didn't find any drastic differences. Plants grown with LEDs had a slightly higher chlorophyll content. But as for leaf morphology, plants grown under red and blue LEDs had lower leaf area, but the leaf anatomy in plants grown under 400 micromoles per square meter per second was essentially the same as in control plants.
And then another question on plant age at harvest. You mentioned some differences in what you call the life cycle and the number of days. Does that mean the weights were taken at different days, or?
Most of our plants, we studied most of our plants at the age of 27 to 28 days, which is the normal harvest age for this cultivar. But some of our experimental plants we left until their natural end of their life cycle. So essentially it was the time when they just dried off and died, despite having sufficient nutrition and lighting.
So they were grown from seeding to harvest at over 27 days?
Yes. Most plants were grown just until harvest.
Thank you.
Still have time for one question.
I have two questions. One question is, this equipment, this facility is for rotating?
This is a model for a space greenhouse designed in our laboratory. It is called Phytocycle SD. Maybe you have heard about it. It's well, according to the design, it is supposed to rotate in microgravity to. It's supposed to rotate in experiments on Earth to simulate microgravity.
It indexes every day you harvest.
Yes.
Okay, I see. Okay, so I'd like to move to the main question. So, do you have any information about the far red light in your experiment? I mean, far red light
Yes.
in the range from 700 to 800 like nanometer range will affect the flowering or sometimes the growth rate.
Yes, of course. In the lighting sources based on red and blue LEDs alone, they did not emit any far red light. The HPS lamp has a slight emission in the far right range, and the warm white LEDs, which we used in our further experiments, they also have a slight emission in the far right range.
We have time for more questions. This is Professor Wu.
mean? What does warm white LED mean?
What about warm white LEDs?
Yeah, warm white.
Warm white?
Yeah, warm white LED.
It's the warm white LEDs are essentially blue LEDs with some fluorescent part added to them, which